Now let's focus on our neurons. Neurons are kind of like the main cell of our nervous system, and they are the ones that transmit the electrical messages. They have a main cell body, which houses the nucleus, and then they have all sorts of processes or fibers that protrude out from that cell body. Dendrites receive signals, and axons send out signals. Axons are going to end in an axon terminal, which will then synapse or connect with another tissue or neuron. So this is a stereotypical example of a motor neuron. So we see a cell body here with the nucleus and some mitochondria. These smaller processes are dendrites, and then it has a long, skinny process called an axon, ending in an axon terminal. Here, Schwann cells form the myelin around the axon, and we can see that the signal will travel in from the dendrites down the axon, ending at the axon terminal. Now, these axons are in some cases very long. Think about, you have an axon that comes all the way from your spinal cord all the way to your big toe. That's a long distance to cover. And again, that myelin helps speed up the signal so that it can reach your toe in an instant. Let's first define some vocab words, which I know is everyone's favorite, and then we'll talk about our action potential. Okay, coming at you some words. A nuclei is what we call not the nucleus of a cell, but in neuroscience, a nuclei is a cluster of cell bodies that we find in the central nervous system. Usually they have a defined function. So then we have a nuclei responsible for motor function, for example. The same thing in our peripheral nervous system, we call a ganglia. Now, the reason for this divide is that there were researchers who were studying the central nervous system and some studying the peripheral nervous system, and they came up with different words, and they became established as different words, but they really mean the same thing. So nuclei, cell bodies in the central nervous system, ganglia, cell bodies in the peripheral nervous system. A tract is a bundle of neural processes in the central nervous system. And it's the same thing as we call a nerve, a bundle of processes in our peripheral nervous system. Now in our brain, we have white matter and gray matter. And white matter is the collection of those myelinated processes, those myelinated axons. And they look white because of the fat. So if you think about a raw steak, which is vegetarian, gross. But that raw steak has a white fatty part, and that fatty part, again, is white. It's that white outer layer. Apparently it makes steak taste good or something. So we call that in your brain too. So the white area is the fatty myelin that surrounds your axons. And the gray matter is the unmyelinated processes and mainly the cell bodies. Okay, reminder about some words. Afferent are the sensory nerves. They approach the brain, and efferent are the motor nerves. They leave the brain. So the afferent ones have their cell bodies outside of the central nervous system, and the efferent ones have their cell bodies inside our central nervous system. And we also have interneurons, which are like short connector neurons. Okay, let's talk about the meat of it here. So I want you to think back to muscle cells. What happens to the muscle cell when acetylcholine binds to the membrane? Well, it opens up that cation channel, right? And sodium will come in, and that will lead to a depolarization. The electrical signaling that happens on neurons is similar. So like muscles, neurons have a polarization to them. They have an electrical potential. And what that means is that there is a separation of charge. The inside of our neurons is negative, and the outside is more positive. Keep in mind, sodium is positive. It's mainly on the outside of that membrane, and it drives this membrane potential, or the difference in charge across the membrane. So a neuron is going to receive a stimuli, and it depends on where it is. So if we're talking about our eyes, the stimuli to our eye is light. Our ears, it's sound. Our skin, it's pressure. It could also be a chemical signal from a nearby neuron. It's going to get some sort of stimuli that are going to open up some cation channels. And these cation channels in the neuron are a little different from the ones in the muscle. They're specific. We have a sodium channel and we have a potassium channel. In the muscle, it was like a mixed bag cation channel. We really just talked about sodium. Here in the neuron, we got a separate sodium channel and a potassium channel. And these two channels have different speeds at which they operate. Sodium, this is our sprinter, going to open fast. Potassium is our marathon or a long distance runner. So let's think about what happens to sodium first. Voltage gated sodium channel is going to open. So that stimuli is going to open up these voltage-gated sodium channels. And sodium, based on its concentration gradient, because there's so much sodium outside the cell and so little sodium inside the cell, sodium is going to rush in to our neuron, and it's going to depolarize that membrane, lead to a positive charge in our membrane. Like any good sprinter, though, it can only do this for a short period of time, and afterwards, it's going to need a break. This is called our inactivation phase. So the sodium channel opens, sodium rushes in, and then the sodium channel inactivates. It's like, excuse me, I need some water, I need some Gatorade, I'm done. Okay? So sodium opens, inactivates. And this will move down the membrane. So if we start with that stimuli, it opens up those sodium channels. Sodium rushes into our cell and depolarizes our neuron. That depolarization is the signal for nearby sodium channels to also open. So sodium will rush in, and then nearby ones will be like, hey, wait a minute, there was a change in voltage. There was a depolarization. I too am going to open, and they will open. And in that way, it'll propagate down our axon with channels opening and opening and opening and flooding in more and more and more sodium. Now, this moves only in one direction because the channels inactivate. So the channels that are open will soon be inactivated and therefore cannot respond to the depolarization. So they open, they inactivate, and then they close. There's a second phase to our action potential. So the action potential has two phases. It has the depolarization driven by sodium entering the neuron. The second phase is by potassium. It's called the repolarization phase. So the potassium channels will open slowly, and then they will leave the cell based on its concentration gradient. So there's a lot more potassium inside of our neuron. So when you open up those potassium channels, the potassium will take that positive charge and it will leave our neuron. And this returns the membrane to a negative charge on the inside. So we can think of the action potential as a moving wave of positive charge coming in with sodium and then leaving with potassium. In with sodium, leaving with potassium, and that will move down our axon. So that action potential, again, is that movement of electrical charge, the wave of positive coming in, positive going out. Now, at the end, all of our ions are mixed up. We got sodium inside, we got potassium outside, everything's a hot mess. And so our cell will burn a lot of ATP to use a sodium-potassium pump to put all the ions back where they belong. Sodium outside, potassium inside. Now, what happens when the action potential reaches the axon terminal or the synapse area? So this is an electron micrograph showing you that synapse. We've got a presynaptic nerve terminal full of vesicles, which are those little membrane circles, membrane bubbles. And we talked about this already when we talked about muscle contractions. Think back to that motor neuron. The action potential arrives and it opens up voltage-gated calcium channels. Calcium will enter the axon terminal and cause the release of vesicles full of that neurotransmitter, which will cross our synaptic cleft and land on receptors on an adjacent neuron or an adjacent target organ, like our skeletal muscle. So calcium is responsible for changing that signal from an electrical signal to a chemical signal. Now, those neurotransmitters bind to a whole bunch of different types of receptors and have all sorts of responses, but one of them is to trigger an action potential in that nearby neuron. That could be the stimuli for another action potential. Again, this is electrical chemical signaling. The chemical part occurs in the axon terminal with the fusion of vesicles causing the release of neurotransmitters into that synaptic cleft, binding to receptors on the receiving neuron's membrane. It can then open up ion channels and cause a signal. I want you guys to pause this video, or as the video ends, draw it out. Now, I know sometimes people get very frustrated with the amount of pictures I asked you to draw, and that's because I really believe that pictures are a great way to learn. So, end the video and try and draw out the steps of an action potential. How does it start and how does it spread down the neuron?